This is AP Bio, and this is our review of Unit 4, and that's all about cell communication. And I want to remind you that there is only one specific chapter that the College Board insists that you need to talk about in cell communication. And what is that topic about? Cell cycle. Yeah, it's mitosis and the cell cycle. Oh, okay. Yeah. And so all the other things that we discuss in here, when we were using hormones in the endocrine system, it's already freaking out. We're using hormones in the endocrine system and homeostasis and feedback control, all of that, immunity and plant signaling. We're using those as illustrative examples of cell communication. Okay, and those are things they might ask you to apply that information to. But the one thing you for sure need to know is you need to know the cell cycle, mitosis / cancer. All right, so first things first, on this one, can you answer the following? Look at chapter 40 about the endocrine and exocrine system. Put myself up here. Gentlemen, tell me if I'm out of the frame ever, because I can't see anything to know. I have no idea where I am. Okay, did you read through those? Now, once again, where it says, did you do your highly suggested reading and thinking? For the final, the only highly suggested, no, because I'll put it in the quizam. There will be no highly suggested reading and thinking on the final. So you could just like take that off your plate. Okay. But that's here because I would use this review when I was reviewing during the regular school year. Does that make sense? Okay. So as we go through this, first off, hopefully you've taken a moment to look through that. So it moves and then it doesn't move. Where's my one? Okay. Sorry, delay. Okay, so one of the first things we talked about was exocrine versus endocrine glands. And to differentiate those, remember that endocrine glands will secrete into your what? Bloodstream. An exocrine gland will secrete into a what? Perfect. Okay. And good. That's good on that. Okay. Next, we looked at different types of signaling. So remember nervous versus endocrine system, right? Which one is faster? Right, because it travels a shorter distance with the chemical signal. It's a very, very short distance right here across the synapse. Whereas when we look at most hormones, they are traveling at a distance within the body. What do you call if it travels a distance between two different organisms? Perfect. Those are pheromones, right? Okay. So we looked at some like unusual situations, and I have the diagram here where you look at the hypothalamus and the pituitary. Because let's just review blood flow one time. Put your hand over your heart. Here we go. Arteries go away from the heart. Veins go towards. Arteries, arterioles, capillaries in the middle, venules, veins. So the only way you can put something in or out of your blood is at a what? Capillary. So when we look at the hypothalamus, which is full on nervous tissue controlling the pituitary gland, the pituitary gland is known as the what gland? Master gland. Why is it called the master gland? Yeah, it controls so many different things. So you want that to be fairly speedy, because if the hypothalamus put hormones into the blood supply at the capillaries, they would have to come all the way back to the heart, right? Out to the lungs for your pulmonary circuit, back to your heart again, and then out to your systemic circuit. Those hormones would be greatly diluted. They'd still work, but they'd be greatly diluted. It would take a little bit of time and then impact the pituitary gland and what it secretes. So one way to do that is to have not one set of capillaries, but two. So the hypothalamus will put, for relating to the anterior pituitary, right? Because the anterior pituitary, did it evolve down out of the hypothalamus? Who did? Posterior pituitary. Remember, the pituitary gland kind of looks like looks like testes. I don't know. I always get afraid on YouTube what I can say. So the hypothalamus goes directly, but Tamir laughs, so I know I could. So hypothalamus goes directly down into the posterior pituitary, direct connection. And in fact, the hypothalamus, full-on nervous tissue, makes the hormones of the posterior pituitary, yes? But for the anterior pituitary evolved up and out of the what? Mouth. And so you don't have that direct connection. So the neurosecretory cells only extend as far as the what? Stalk. And in there, they put their releasing hormones into the stalk. Those are then carried by the blood to the anterior pituitary. They come out of the blood at the anterior pituitary and tell the anterior pituitary what to secrete. So then it puts its stimulating hormones into that second set of capillaries, which does go back to your heart, to your lungs and your pulmonary circuit, back to your heart, and then out to your systemic circuit. And I do have a slide to show you that, but I wanted to include that when you see right here where it says neurosecretory cells, it's not really a gland, it's not really a nerve cell, it's kind of an in-betweener. Okay? Then we had a slide here just comparing the speed of the nervous system with the endocrine system. One thing you want to remember is that the endocrine system's effects are usually very what? Short lasting or long lasting? Long lasting. There you go. And I'll let you compare and contrast that. Okay, then part of that cell communication was comparing and contrasting both positive feedback and negative feedback. So positive feedback, the response that you get in the positive feedback, like positive feedback is saying more, 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 right? So that positive feedback response increases the stimulus. So now you have more stimulus, now you have more response, and this is like a little circle. And then until you get to some climactic event, right? And the examples, what is the like classic example for that? Giving birth. Also, what's another one I gave you? Yeah, blood platelets and clotting. That's another example. There aren't a lot of examples of positive feedback. There's one in the menstrual cycle for females, but there's not a lot of examples there. Most feedback is not positive, but negative feedback. The response to the stimulus decreases the stimulus, right? It's getting it back to, what do you call it? Okay, I'm looking for something else. A set point. It's getting it back to a set point, returning it. It's kind of like cruise control on your car, right? And so there's different ways to achieve that. Another one that I didn't put in here to keep you at a set point in order to maintain homeostasis was contrary hormones. Do you remember that? And what's the classic example for contrary hormones? Insulin and glucagon, right? Because insulin does what? Lowers blood sugar, and insulin raises blood sugar. Perfect. Insulin lowers blood sugar and glucagon raises blood sugar. Sorry, I'm not even thinking. I got up at 3 this morning. 3. It's a long story. We'll save it for a different day. Yeah, you went to bed at 3? 12:30, yeah. And I've had meetings since 7:30, including through lunch. I have not stopped. I like inhaled my lunch. I don't even think I actually ate it. I just sucked it into my body. Okay. So here we can see an example of positive versus negative feedback about the clot growing. And then here with negative feedback, it's self-corrective, right? You're trying to get it back to that set point. All right. Then next we need to talk about the way hormones are able to communicate with the cell. So we had steroid hormones versus what? Steroid hormones are lipid soluble. So steroid hormones versus water soluble or protein hormones, right? And so steroid hormones, because they're a lipid, they can go right through the phospholipid bilayer, right? They go through it. And so the receptor for steroid hormones is internally to the cell. You could have a receptor out here in the cytoplasm. Oftentimes it's impacting what DNA is transcribed and translated. These are often your gonadotropins, things that are affecting reproduction, right? Whereas your protein hormones, they cannot go through the phospholipid bilayer, so they have to bind to a what? Receptor. And this is where we see that example of signal transduction, right? This triggers this, triggers this, triggers this. It might have a kinase that's going to phosphorylate something to either turn some sort of enzyme on or off, and then you end up getting your product, whatever that is. Okay, and then this is where I inserted a little bit about the posterior pituitary and the anterior pituitary. So I'm just going to reiterate the posterior pituitary here, okay, the neurosecretory cells can reach all the way into the posterior pituitary, and the hypothalamus makes the hormones the posterior pituitary secretes. It stores them. Then another set of neurons actually reaches into the posterior pituitary and tells it, now release these hormones now. Whereas your anterior pituitary, again, the neurosecretory cells only reach as far as the stalk. They are the releasing hormones are put here in the blood supply. It travels. You have a second opportunity at capillaries where the releasing hormone comes out and tells the anterior pituitary which stimulating hormones to release. And so if you look here, one more, you can see those. And one of these you recognize right away, oxytocin. What is oxytocin? That's the hormone that makes you what? Contract the uterus in order for a baby to be born. That was our classic positive feedback example. As the baby's head, hopefully head, not feet, but as the head push against the cervix, that sends a signal back to your brain and it tells you contract even more, right? And so then the hypothalamus tells the posterior pituitary secrete even more oxytocin, which increases the contractions, which makes you push even harder against the cervix. And ultimately, you're going to be brought back into homeostasis, right? Because eventually you're going to get that baby out of you, right? And then you're brought back into homeostasis. It's just you're going out, you're getting there the hard way, right? And then, whereas the anterior pituitary, you're looking at all these types of things. This has to do with like either reproduction or feeding the baby or growth or metabolism. So you combine these two, and ADH stands for antidiuretic hormone, impacting your kidneys and water reabsorption and the collecting duct. All of this has to do with maintaining homeostasis. That is why it's called the master gland, and it influences so many other glands. Okay. Next, when we talked about, okay. Dun, 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 dun. Let's go here. There we go. Second messengers, right? And we saw cyclic AMP was one that was used several times, right? We talked again about that signal molecule. What kind of hormone would this be if we're using a second messenger? A ligand is anything that's going to bind, but tell me what kind of hormone might this be? A what kind? A water soluble, a protein hormone, right? Because it can't come in. And it has a receptor, which triggers a G protein, which triggers adenylate cyclase, which makes ATP become what? Cyclic AMP, right? And then that sends off, that is your second messenger. But we also saw other second messengers like calcium being a second messenger, right? That was something that was kind of universal. And so this initial communication, right? Because this is all underneath the umbrella of cell communication. That initial signal, that initial message never actually goes into the cell, but it does have an impact on what goes on inside that cell. Okay, moving on. I'm going to go to, okay, chapter 9. This is the have to in this unit. It's about cell cycle. It's about regulation of the cell cycle, mitosis, and then when things go bad, when we talked about cancer. Okay, so just take a quick minute to look through that. You learned a lot this semester. All right. So on your cell cycle, okay, we know that the bulk of the cell spends its time in what? Interphase. And we know interphase is broken down into how many substages? 3. What's the first one? What happens during G1? Growth, and you make more organelles, and also it's a checkpoint, right? Did mitosis go well, right? So growth gets bigger, more organelles, and we check. Okay, what follows G1? S stage. Oh, did everybody sign in today? Is there anybody who still needs it? Okay, sweetheart, will you just put on that back table for me? Thank you, thank you, thank you. So S stage, what does that stand for? And what happens? What gets formed there? Be real specific because you know now. Sister chromatids. Do sister chromatids only separate in meiosis? When else do they separate? Anaphase of mitosis and anaphase. That's right. Say it. Anaphase 2, right? Does that make sense? Think it through, right? You will separate those sister. Yeah. Yeah. Back up. You will remember in mitosis, you line them up single file, right? And you just separate those sister chromatids. In meiosis 1, you separate the homologous pairs. And in meiosis 2, you separate the sister chromatids. So both in mitosis and meiosis, those sister chromatids will be separated that you formed during the S stage. What follows S stage? G2. What happens in G2? Make a bunch of proteins. Good. What follows the G2 stage? Say another name for me. M stage, right? That M could be mitosis, or that M could be what? Meiosis, which you're learning in your current unit, right? Okay, it's just M. When you say M, like for in this case, if we're doing mitosis, that M will be mitosis and what? Cytokinesis. Okay, let's go over the stages real quick. The first stage is called what? Sorry, something's in my hair. Something's in my hand. Okay, sorry. The first stage is called what? Prophase. The chromosomes become visible, and they are what? Sister chromatids. Good. Just curious, from the current unit, what would you call these? And if they had not done the S stage, would these still be homologous pairs? Okay, good. The chromosomes become visible, the nuclear envelope and the nucleolus disappears, radiating microtubules, asters. Now, what does an animal cell have in the middle of its asters? Centrioles. What does a plant cell have? Nothing. Okay, good. Asters. Then they meet in the middle at metaphase, away at anaphase, to nuclei at telophase, cytokinesis. Those daughter cells that you form, they are exact replicas of the parent cell, right? One parent cell, how many daughter cells? Even for men and women? Yeah, the only time you have that unequal is when you're doing meiosis, right? Okay. Next, we talked about that cell signaling. Do you remember this series right here? Remember we talked about the cyclins, right? And there are more cyclins than even what is displayed here. But we know that those cyclins, they bind to CDKs. Do you remember what those are? Exactly. Anything that influences your cyclins or your cyclin-dependent kinases is going to influence your cell cycle, right? Anything that influences those two. And we talked about our example of how the cyclin binds to the CDK, then the CDK can phosphorylate some other protein, possibly turning it on, but also possibly turning it. There are options, right? There are options. Okay. Then next we talked about, did I skip over a page? No, I didn't. Apoptosis. Is that a normal part of life? Yes, it's controlling our cell numbers. In the immune system, where did we see apoptosis? After what? After a battle, but after you've done the amplification of that specific B or T cell, right? Who have fought the fight. Remember, you make that clone army, right? Of B cells that recognize that particular pathogen. And so once you've won that battle, then those cells are all going to commit what? Apoptosis, except for what? Memory cells. You don't get that amplification of your innate immune response, right? The natural killer cells, the dendritic cells, they don't learn. You don't get more of them when you fight that infection again because they don't know any better. All they know is stranger danger, right? They just know if something is foreign. Okay, good. But what I think you're getting at there is about those natural killer cells, how they will dock with a cell. That's how they will kill it, is in an apoptosis manner. All right. So then we talked about, and there's P53. You saw that in there, right? Okay. All right. Then we just talked about the stages. We just went through all those stages. So I think you feel good. I wouldn't color it out like some of you are doing, but yeah, there you go. Okay. And then here are all those stages. Now, cytokinesis is different if you are a what or a what? Anything that has a cell wall would do it differently, right? Because they have to rebuild the cell wall. Perfect. Okay. And then bacterial cells do not do mitosis. Prokaryotic cells do not do mitosis. Whereas eukaryotic cells have linear chromosomes and they are diploid, right? Prokaryotic cells have a single circular chromosome that replicates, attaches to either end of a cell wall, and then it separates. And that is called binary fission. What else does binary fission besides prokaryotic cells? Yeah, mitochondria and that is evidence for the, see how smart you are. You're so smart. Okay, then where am I at? There we go. So then we talked about what happens when things go bad, right? And we said normal cells exhibit a couple of things, right? Contact and anchorage. Good. Now there's some cells who don't have that, like blood cells, lymphatic cells, right? Because they're floating all around there. Okay. But normal cells have those things. When you lose that ability where you can just go anywhere and keep multiplying and you're not supposed to, that's bad because you can set up camp somewhere else. You don't detect crowding, so you over reproduce. And so we talked about those growths and neoplasm or cancer. If that cancer cell is encapsulated, what kind of tumor do you call that? Benign. It's there. It's too many cells, but you could just go in and get it out. Malignant is when it has spread to a different area. Okay. Now, sometimes these tumors, they just run out of nutrients, right? They're overgrowing, but they're still enclosed. What do you worry about with that? Yes, angiogenesis, which is when they secrete through cell signaling, they tell blood cells grow here, right? So that's a bad signal, right? They're saying, come over here, give me some nutrients, give me some oxygen, take away my waste. Then they could get even bigger and maybe they could live longer and gain a mutation in order to escape wherever they're being compartmentalized. And we talked about having two genes, okay, that are super, super important, okay? Proto-oncogenes, normal, right? Normal, but the reason why we worry about them is because they're in a position of power, right? So if they go bad, they can influence a lot of things. And what they do is proto-oncogenes, what do they do to the cell cycle? They promote it and they inhibit apoptosis. Okay, that's what they do. Where conversely, tumor suppressor cells, if they're doing their job, any cells that are out of control, proto-oncogenes becoming oncogenes, then they do what? Yep, they do apoptosis. And I gave you some classic examples of both of those. The RAS gene is normally should only be triggered if you had a growth factor, a growth hormone binding to a receptor triggering the RAS, right? Saying, we're growing now, please make more cells. This is a RAS that is not getting triggered by a growth factor binding to a receptor, and it's stimulating that cell cycle. And we also talked about the breast cancer genes being examples of that as well, okay? And then over here, tumor suppressor genes, we talked about specifically one we want to be concerned about is P53, right? Because P53 makes a cell, if it is transcribed and translated correctly, it will make a protein that will do what to the cell cycle? Stop it, right? And then you'll try to what it? And if you can't fix it, then you kill it. Exactly, using caspases. So that's why it's so important. Okay, then that brings us here. Chapter 33. Skim through that bad boy. All right, that's all the time I got for you. Let's keep going. So you really cannot talk about the immune system and how it functions without having like a basic understanding of the circulatory system. And we've already talked about that a little bit because we've talked about all exchange takes place at the what? Capillaries. And the key thing about the capillaries that we focused on are the two opposing pressures. Think for a minute. Got them in your head? Okay. So what is the one when you approach a capillary bed? What are you in? What kind of vessel are you in when you approach a capillary bed? You're coming into it. Arteries, arterioles, capillaries in the middle. So you're at an arterial, right? And so you're assuming that the blood pressure known as the hydrostatic pressure is going to be greater at the arterial end of a capillary bed as opposed to the what? Venular end of the capillary. And the reason is because you're farther away from the heart, which created that pressure. Do you agree with that? Okay. So when you go into a capillary bed, that hydrostatic pressure is forcing things out. And how does it get out? In between, remember, a capillary is one cell thick, made out of, what are the name of the cells? Do you remember it? I thought I taught it to you, but I don't 100% remember. Did I teach you endothelium? Endothelium. I'm teaching you now. Okay? Endothelium. Those are the cells that actually line everything of your circulatory system. It's just by the time you get to the capillary bed, that's all that's left. Okay? And that is forcing fluids out. Also, there's a little bit of this going on. What's this? Yeah, endocytosis and exocytosis. And there might be a little bit of this going on. Carriers, right? But the hydrostatic pressure is greatest at the arterial end, forcing fluids out. Still in there, what's the opposing pressure? Osmotic pressure, right? And so you're going to go, water must. So it's going to want to flow into, right, your capillary bed. But if the hydrostatic pressure is greater, more fluid goes out. But by the time you get to the venular end, the hydrostatic pressure drops and the osmotic pressure is greater. And so fluids will flow in. And that's usually going to be carrying things like waste and carbon dioxide. Okay, you all right with that? Okay. So then we said, hey, what are the functions of your lymphatic system? So there is an unequal amount, an inequity in the amount that gets pushed out at a capillary versus what gets recovered. So one of the key things that your lymphatic system does is pick up all that excess fluid. What is it called if there's failure to do that? Starts with an E. Edema. Yeah, and you get that swelling. Remember that leg? Remember that leg? It's beautiful. Okay. One thing that people forget is the lymphatic system also absorbs fat at the intestines, and a job is to produce lymphocytes. So we can see the connection between the circulatory system and the lymphatic system is when that excess tissue fluid is picked up, it is screened. It has a little border crossing at a lymph node before it gets returned to the subclavian vein. Now, what if you're a toxin that didn't get pushed out? You will get filtered at the spleen, exactly. Remember, your spleen filters blood like your lymph nodes filter lymph. You okay with that? What do you call the fluid portion of blood? Plasma, right? Plasma. And dissolved in there are gases and nutrients and salts and proteins and vitamins and waste and hormones, okay? All of that moves out. If it moves out of the capillary bed, it's interstitial fluid at that point or extracellular fluid. That fluid moves into lymphatic vessels. It's now called lymph. And then it gets returned to the circulatory system. Good. All right. So then we looked at the major organs, right? And the thymus gland, look where it is. Remember, you have little eviscerated girl because your thymus gland gets smaller and smaller and smaller as you age, right? And what's the key thing we want to remember about the thymus gland? What happens there? Good. Okay. Your bone marrow, what happens there? B cells mature, but all blood cells are made, right? Red blood cells, white blood cells, their home all comes from that. But that's where your B cells mature. Lymph node, we already said, filters lymph. Spleen filters your blood. Okay? When you have an injury, okay, you will have a wound. You're enacting your innate immune response, usually, right? What you're born with. And for all of us, that is the same, right? You have a cut, you're going to bleed, you're going to send platelets. We already talked about positive feedback in order to build up those platelets. Who is usually first on the scene if you have a wound? Neutrophils, because they are the most numerous. Yeah, good. Who else could show up? Okay, macrophages. How do they get there? Monocytes, smaller, so it allows them to get out of the capillary, and then they get bigger into the macrophage. Who else could show up? Mast cells. We like mast cells because of what is in them? Antibody, and I think I heard immunoglobulin. And in fact, you know which one? Immunoglobulin E. Good. Immunoglobulin E, just pulled that out. is into the mast cell will start secreting what? Histamine. And histamine starts with a V. Boom, shagalaka, vasodilation, okay, which increases the spaces between the endothelial cells of the cat. Thank you, Nikki, of the capillary bed. He is the fish tank master. I do nothing like this for the fish tank. Okay, so it makes it, and that can cause swelling. Swelling can cause, ouch, what can it cause? Pain. It can also increase the temperature, which then might kill off any bacteria that are there. Okay, what else could you have there? Did we name them? Yeah, you could have eosinophils, go for the worm. But these cells are releasing and pulling a fire alarm. What are they releasing? Well, they can release cytokines, and one of those cytokines can be interleukin, which amps up your entire immune response, right? And maybe if you've already experienced an antigen in there, it might even trigger some what? Memory cells, right? But remember how they are stimulated. Those memory cells remains the same. If it's a B cell memory cell, it has to come in contact with what? The antigen. Has to come in contact with the antigen directly. Whereas if it's a T memory cell, it has to be what? Presented. If it gets presented on an MHC1, that cell will be done. Okay. If it's presented on an MHC2, I will help you. Good. All right. So that brings us here, right? The two types, antibody mediated versus cell mediated. This is called your adaptive immunity because it's literally an adaptation you can get during your lifetime, right? Depending on where you've traveled, then you will have antibodies and memory cells for whatever you have been exposed to. Just make sure you separate the two out. Remember, T cells are going to fight your own body cells that have gone bad. They could even fight a cancer cell. Whereas your B cells do not have to show up to where the invasion is. They just have to release what? And antibodies are very, very what? Specific. Good. Okay. And so then I talked about, let's talk about the orientation of your adaptive immunity. So what's going to happen first, right? A T cell gets presented with an MHC2. And who has MHC2s? Dendritic cells, macrophages, B cells, they all have MHC2s. Okay? When they present, okay, we're not yet green yet because that T cell needs to run into a virgin who has direct contact with the pathogen, right? Direct contact. Then they agree, and then they will both start to make their clone army. The B cells will make two. What are the two it makes? Memory cells for next time, and those plasma cells will secrete, whereas the virgin T cell will form cytotoxic T cells, cell-to-cell combat. What else? Helper T cells to amp up the response. Memory T cells. Good. All right. So then here, I kind of talked about all the kinds of things that antibodies can do, whether it's in a mast cell releasing histamine, triggers, macrophages like to eat, natural killer cells like to kill. Complements, I didn't mention that in the innate immune response. Remember the complement proteins, what do they like to do? Drill holes, right? So those are all things that are there. Whereas your T cells, right? Your T cells, we already just went through all that. Same picture. Yes. Memory B and memory T? Totally. Because a B cell, if it's triggered, if it's a memory B cell, it's going to make plasma cells to secrete antibodies. And the T cell, if triggered, is going to make cytotoxic T cells and helper T cells. Does that make sense? Okay. All right. And then I already talked to you about MHC1, MHC2. You feeling comfy on that? Okay. And then this was the big picture. You have that, the summary kind of, and I just went through that with you. If you need more time there, you have this slide presentation. You can use it. Okay. And then, let's talk about interferon. When one of your body cells is infected, right, it wants to warn everybody else, save yourself. Maybe you should just go ahead and commit apoptosis now. Stop making any proteins because you might be making viral particles. So that is interferon, okay, that gets released. All right. And then, lo and behold, these last 4 minutes, plants. What? Stop, Mrs. Sloan. Stop talking right now. I can't do anymore. Okay, I'll let you read that part on your own. Okay? But there's nothing new here that you haven't already learned in what we've already reviewed today, right? Look at some scenarios right here. You're looking at a carrier going in, affecting gene expression. You're looking, remember, the phytochromes detecting light. Yeah, okay. So you have signal transduction again with defense. You have seen that before. Now, one I want to point out too is auxin, because that is like the go-to hormone for plants that I wanted you to remember. And because auxin facilitates plants responding to their environment, right? Because they are able to communicate to which cells need to get bigger and face the what? Sun, right? And remember, auxin travels to what? The sunny side or the shady side? Shady side, and it gets those cells to elongate. All right. And that's the acid growth hypothesis. Remember, where it puts hydrogen ions into the cell wall to weaken the cell wall so that if water goes into the vacuole and the vacuole expands, it pushes against the cell wall. Okay. Then we also looked at if you have a wound, we can see some signal transduction here. You might release a chemical to call in a predator, right, to help you defend that area. You might make a barrier like a fire break amongst your tissues so that a virus can't go through another living cell to get, kind of saving the rest of the plant. You might release a toxin or a poison to impact that herbivore. And remember, the enemy of my enemy, what? Finish the sentence. Is my friend, right? So then you would call them in. We looked at mutualistic relationships, the classic acacia tree and ants, okay? And then here, let's talk about that light sensitive pigment phototropin. We talked about auxin getting, when it detects where the light is, it moves auxin to the shady side. But the part I want to talk about here is photoperiodism. And that is plants' ability to tell what? Time. Some plants are long day plants, which we know means they need and some are short, which means they need a long night, because it's really the length of the night. Because if you interrupt a long night with a flash of light, then it becomes a short night, okay? And here is the second to the last slide right here. Remember, PR, when exposed to light, it's like a toggle. It slowly changes into a new shape, which is the PFR shape, right? And then as evening approaches and during the night, it switches back to its original PR shape, right? And some plants are neither long day or short day. They are day neutral, okay? And that, my friends, was that unit. I am so proud of you. Good job. Good job. Good job. You are welcome. Oh.